The last IPCC report shows that some of the consequences of climate change are already inevitable. Global temperatures are predicted to rise by at least 1.5 degrees until 2050, regardless of the measures implemented now. Agriculture as we know it faces the biggest challenge in all of history. Higher temperatures, droughts and flooding will result in crop failures around the globe and further stress our supply chains. To tackle these challenges, we clearly need crops capable of withstanding a variety of extreme weather conditions. Despite that, the speed of innovation in crop improvement is far too slow. It currently takes about 10 to 15 years to develop new crop varieties. Something has to change for us to efficiently and quickly develop new crops. This limiting factor is observable in the iGEM competition as well. Out of 1,838 teams, only 37 teams tackled the challenge of working on plants since 2015. But what are the main reasons? Apart from the long generation times, there are also only few resources available, from protocols to genetic parts and other projects you can build on. To pursue our dream of a blooming plant community in iGEM, we teamed up with Bielefeld in a close partnership. We founded a community of 20 teams and hosted 4 meetups on phototrophic organisms. We discussed topics chosen by our community and included dedicated troubleshooting sessions with experts in plants and bio. During our fruitful discussions, we decided to team up with 11 phototroph teams to create a 160-page handbook filled with accessible resources. We hope that this partnership between our teams sowed the seeds for a wider iGEM phototrophs community. Nonetheless, going back to the root of the problem, the inherently slow growth of plants is still a major obstacle. In our project, we address this by establishing cell-free systems from chloroplasts as rapid prototyping platforms for plants and bio. Cell-free systems are in vitro tools capable of transcription and translation. By creating a crude extract from an organism of choice and supplementing it with external resources, such systems are capable of directly prototyping genetic parts. But why do we focus on plant chloroplasts? These organelles offer several advantages, such as high-precision engineering via homologous recombination, the absence of transgene silencing, transgene stacking in synthetic operons, and high-level expression of gene products. In addition, chloroplasts are inherited maternally and thus not passed on through pollen. Their targeted engineering opens up new dimensions of breeding safer genetically modified crops, reducing the probability of outcrossing with a local flora, an important step to address major ecological concerns from the public. So how did we go about creating our cell-free systems from chloroplasts? Our system consists of 3 main components: the crude chloroplast extract, a reaction mixture, and the desired DNA construct. In our workflow, we obtain the extracts by isolating intact chloroplasts and subsequently lysing them to obtain the crude extracts. Intact chloroplasts were isolated by blending fresh leaf material and performing density gradient centrifugation. This method allows us to specifically continue our work just with these organelles. In the following step, we lyse the chloroplasts by passing them through a needle, and the resulting shear forces break open the organelle and release its components. The destroyed membranes are collected by high-speed centrifugation, which leaves us with a chloroplast extract that contains all necessary machinery to perform transcription and translation reactions in vitro. Of course, there's much to be varied, and we put a lot of effort into getting it just right, so that it works in as many plants as possible. We went through several rounds of optimization. To boost expression levels in our system, we had to improve the yield of our chloroplast isolation and had to perfect the lysis. Another crucial factor for us is the cost and accessibility aspect. By using sucrose for our density gradients as an alternative to percol, we were able to lower the costs of our protocol by an order of magnitude. This would make this technology accessible to future iGEM teams and allow them to develop their own extracts of every plant species they could dream of. We managed to end up with a protocol that allows us to reliably obtain chloroplasts from tobacco, spinach, wheat, rice, maize, soy, tomato, and even oak leaves. Sadly, as growing enough biomass from all those plants proved difficult, we had to focus on a smaller subset of species.
We can proudly say that through these efforts we developed highly active chloroplast cell-free systems from tobacco, spinach and wheat. What you see here are measurements from our luciferase reporter nanolook in arbitrary units. We compared nanolook expression in chloroplast extracts of tobacco, spinach and wheat, showing that they are all highly active compared to the controls. But the expression wasn't that high from the beginning. The reaction mixture would see a lot of improvement. Most crucial components here are the magnesium and potassium salts, which is why we tested a range of concentrations to find the optimum for highest expression. In addition, titration experiments were performed to assess the influence of varying DNA concentrations on gene expression levels. These first rounds of optimization allowed us to settle on a reaction mixture with the most reliable expression. Now we aim for optimizing the last missing piece of a cell-free reaction, the DNA template. We created heavily optimized universal test construct to screen for expression in our extracts from different species. It was assembled using the modular cloning approach, where standardized parts can be combined into transcription units. This cloning approach allowed us to apply the engineering principles of standardization and decoupling, which enabled us to test different parts of our test construct individually. First, we analyzed the effect of two 5' UTRs from literature on the expression level of our construct. We could show that the gene 10 UTR from the E. coli T7 phage possesses way higher expression than the RBCL UTR from tobacco. Next, we focused on optimizing the codon usage of our reporter gene. We were surprised with the fact that the nanolook optimized for Chlamydomonas performed better compared to the one of tobacco. This prompted us to use the Chlamydomonas version in our next universal test construct. Finally, for the 3' UTR, we went through several iterations of the DBTL cycle, which allowed us to identify the most efficient one, the tobacco mosaic virus UTR. With this data at hand, we built the 7th and final version of our universal test construct. The 7 rounds of designing, building, testing and learning made it the most optimal construct for chloroplast cell-free prototyping. Having our universal test construct 7.0, we wanted to use it for something really ambitious. How exciting would it be if we could develop a cell-free system from the chloroplast of oaks, a native European species which is threatened by current extreme weather events? Designing, building and testing biobricks for a tree seems impossible in the time frame of an iGEM project. That is why we made it our duty to establish a cell-free system of the English oak. We could show that all of our optimization efforts bore fruits and led to successful gene expression in our oak cell-free system. This allowed us to optimize its reaction mixture, making it possible to characterize even endogenous oak parts. Out of our 157 parts, these are my favorites.
The second big branch of our project was to design, build and test a part collection for use in the chloroplast chassis. This effort was inspired by the severe lack of chloroplast genetic parts in the iGEM registry. Previously, there were only 18, none of them characterized. Now we have expanded this number of available parts to 175. They mainly cover regulatory parts, which are vital for the engineering of more complex genetic circuits. To make them even more useful for the community, we assembled them in the PhytoBrick standard. PhytoBrick is a MoClo standard agreed upon by the international plant community, enabling easier exchange of genetic parts between labs. The parts in our toolbox have been rationally designed by using transcriptomics data and our newly created chloroplast genome database. This database consists of more than 6,000 plastid genomes with 2.4 million sequence features, and it helped us tremendously to identify suitable regulatory elements. As we think that part characterization is the key to establish a valuable toolbox, we set out to test our parts in an agriculturally relevant crop. From these results, we can learn how parts from different species perform. For instance, we can see that the Rubisco 5' UTR from different species behaves differently in the same wheat extract. Testing this in vivo would have been impossible since there's no protocol for wheat chloroplast transformation. With OpenPlast, we could get expression data in a matter of hours. After establishing our cell-free system in a crop, we focused on comparison between species. To this end, we performed slightly different measurements than before. We used a ratiometric approach utilizing two different luciferases. The transcription unit for the firefly luciferase always stays the same, and it's used as a reference point. The part to be characterized regulates the expression of nanolook luciferase. By calculating the relative difference between both, we can reliably compare parts in different extracts by using the ratio as a unitless number. For our first comparison, we measured 10 different 5' UTR in spinach extracts and were able to observe significant differences in the expression levels. In parallel, we measured the same constructs in a tobacco extract, yielding similar expression levels. To highlight the similarity between both even further, we performed a linear regression which shows a high correlation of the expression levels for the different species. These results further cement the transferability of chloroplast genetic parts in different species. Developing our project, we noticed that cell-free systems have already been around for quite a while, which is why we started to wonder why nowadays such systems are not very prominent in chloroplast research. To clarify this issue, we interviewed two leading researchers in the field, Michael Jewett and Lauren Clark. The key to building genetic circuits in a chloroplast cell-free system seems to lie in endogenous promoters. Their usage has been limited though, since they do not promote expression as strongly as the T7 promoter. After our conversation, our next goal became clear. We wanted to improve the system enough so that we get high-level expression using only endogenous parts from chloroplasts. Our efforts paid off. By further optimizing our experimental procedures, we managed to develop cell-free systems fully capable of performing protein biosynthesis using endogenous transcription and translation. This allowed us to characterize different endogenous promoters from different species in our spinach extract. After characterizing all these endogenous regulatory elements, we asked ourselves if it would be possible to design, build and test entirely synthetic parts which would facilitate gene expression in chloroplasts. This is especially of high interest, as using parts twice might cause problems due to unwanted homologous recombination. Here we show expressions of successful, entirely synthetic 5' UTRs. They allow expression of our nanolook luciferase and even vary depending on the UTR design. To benchmark the prototyping capabilities of our cell-free systems, we tried to supplement linear DNA as a template. We were amazed that it worked, and it motivated us to optimize our workflow even further. In the end, we could go from in silico design to experimental data in as little as 7 hours.
A project as ambitious as ours would have certainly been inconceivable without automation. Recognizing the privilege of having access to such exceptional technology, we teamed up with Paris Bettencourt to think about ways on how to grant other iGEM teams access to such automation platforms. In this partnership, we have explored workflows that could form the basis for a globally distributed biofoundry, by iGEM teams for iGEM teams. To put this ambitious vision into practice, we focused on Golden Gate cloning and tested the resulting standardized protocols in an extensive interlab study. And to further support the iGEM community, we have taken the unique opportunity to help shape the iGEM distribution 2.0, both by examining the existing distributions and proposing an alternative, all-encompassing Golden Gate kit to the iGEM registry. One experiment we couldn't speed up with automation though was our ambitious goal of comparing actual in vivo data with our in vitro data of tobacco. A tremendous challenge had to be overcome for us to receive transformants in the limited time frame of an iGEM project, a challenge so difficult in fact that to the best of our knowledge, no iGEM team has ever done it. We used 4 different constructs for the transformation via biolisting bombardment, which are equivalent to constructs used in our cell-free expression analysis. After 8 weeks of regeneration, calli were formed from potentially transplastomic cells. We demonstrated the presence of fluorescent chloroplasts, confirming the first successful chloroplast transformation of an iGEM team ever. By maintaining the selection pressure over the next months, the fraction of transformed chloroplasts will rise until homoplasmy is achieved. Even with the heteroplasmic callus, it was possible to observe nanologue expression and confirm that our construct was successfully integrated. Thus, we are working towards validating our cell-free measurement method. Producing in vivo data was an important step in making our extract attractive to the industry. Another experiment that from our interviews with Corteva AgriScience and KWS seemed to be of great importance was using OpenPlast to screen for new selection markers. For them, the lack of appropriate selection markers is currently the biggest barrier for wider investment in transplastomic crops. So we were compelled to compare the effects of different antibiotics in our system. The inhibition of gene expression is visible in most antibiotics. The high expressions you see are the positive controls. We also included spectinomycin as positive control for wheat due to its natural resistance to it. We were happy to see that OpenPlast realistically responds to the effects of antibiotics and thus can be used to screen for new selection markers. We hope the possibility of easier transformation paves the way for the development of transplastomic crops.
We are very much aware of the controversial status genetically modified plants have. Germany, for instance, has currently no GM crop approved for cultivation, and 79% of the population believes it should stay that way. From our conversation with scientists and science communicators, we identified the need to clarify major misconceptions about the biology of genetically modified organisms. Firstly, we aim to initiate a conversation with farmers from whom we got the impression of uncertainty and hesitancy rather than the resolute opposition we expected. In order to elucidate these open questions, we created this informational booklet with facts about the history, use and problems related to genetic engineering in modern agriculture. To bring this topic to a younger audience, we opted for a learn-by-doing approach. We made use of a cell-free educational kit based on cell-free systems called BioBits. Here we can showcase another advantage of the improved biocontainment provided by cell-free systems. It allowed us to demonstrate transcription and translation outside of a BSL-1 lab. This opens up a series of possibilities of using them in an educational setting. To spread this approach to a broader audience, we talked to the German Association for Synthetic Biology. Together, we will launch a pilot project that will bring cell-free systems and SynBio education to schools around the whole country.
In summary, we were successful in creating cell-free systems from a diverse set of plants. We demonstrated how they can be used as a prototyping platform for the characterization of genetic parts in chloroplasts, even in non-model organisms like wheat and oak. We also consulted with leaders in the industry and performed experiments that showcase OpenPlast as a viable solution to speed up transplastomic plant development. One of these experiments being the first successful chloroplast transformation in the iGEM history. From now on, teams will be able to use our protocols to characterize and use genetic parts for the chloroplast of any plant, be it a model organism, a new food source, or a native endangered species. By lowering the time investment needed for prototyping in the chloroplast, we provided fertile ground for synthetic biology to create the crops that feed the future.